So good day to everyone. This is Dr. Anup Ashok from the Department of Chemical Engineering here at PV Institute of Technology, Narsapur. So I will be continuing with my course on bioprocess engineering, where I will now be talking about the next stage or the next step of the bioprocess engineering. So here I would like to talk about what is an enzyme and what the enzyme's uses are and how the enzyme is put into different activities. So here in this today's topic, we'll be discussing about the kinetics of the enzyme catalyzed. So to begin with that, we'll first talk about what is an enzyme. So enzymes are most commonly protein-based compounds which have high molecular weights. So when I say it is as high molecular weight, we're talking about molecular weights as high as 15,000 to over several million Daltons. So Daltons are the measurement that is used to analyze and determine what the molecular weight of any particular compound or a material is. And moreover, what we would like to say is that enzymes are materials or compounds which acts as catalysts. So you have a chemical based catalyst. So similarly, enzymes are bio based catalysts. So there are certain kinds of RNA molecules recently have shown some catalytic activity. So, but most of the RNA molecules that we are talking about have cellular reactions, and they are basically mediated by some protein based catalysts. So even though the RNA are part of a compound which involves in the catalysis material, the proteins are the basic compounds which helps in analyzing the process and helps in the catalysis of any particular process. So RNA molecules which specifically have the catalytic properties, those are basically called as the ribozymes. So why they are called as ribozymes? So if you know from your basic biological aspects, the site of all biochemical reactions or all the reactions that is taking place in the cells happens on the ribosomes. So similarly, all the reactions are taking place on the ribosome. So thereby, whatever the RNA molecules that are present, those molecules which take part in such kind of reactions at the cellular level, those are basically called as the ribozymes. So one important characteristic of the enzyme is that the enzymes are highly specific in nature, and moreover they are very versatile and also very effective biological catalysts. So what we talk about the specific nature, and moreover we will like to say that it is very specific. So it is estimated that a particular enzyme will only catalyze a particular type of a reaction. So to catalyze any one reaction, only one particular enzyme is possible to do that. No other enzyme can be used for the same process. That is how specific an enzyme can be found to be. So till date, if you see, there are more than 2,000 different enzymes that are known to us. And if you also observe that the rate of reactions involving an enzyme or a biocatalyst is comparatively much higher than your chemical-based catalytic material at ambient conditions. At higher temperatures, yes, there is a possibility that your chemical-based catalysts have a higher possibility or a higher reaction rate than your biocatalyst. But at the ambient conditions, if you look at the biocatalyst or the enzyme-based compounds, whatever we have, those have the highest precedence over any other type of catalytic material that is available. So usually if you name an enzyme, the enzyme is named by adding an additional suffix which is ASE to the type of compound that it is catalyzing. For example, if the enzyme is catalyzing a urea based compound, then it is called as the urease. So that is why we have added ASE to the end of urea. And the other possibility is that it can be named after the process or the catalytic process that it is catalyzing. For example, if it is catalyzing the dehydrogenation process of the alcohol, then that particular enzyme will call it as the alcohol dehydrogenase. So that is usually how any of the enzymes names are coming into existence. So this is the structure of the enzyme. That is a common structure of an enzyme that you see on the right hand side of the figure. So you have an N-terminal domain, a C-terminal domain. So it's a very complex structure of an enzyme. So when we're talking about several million Daltons in molecular weights, you have such a complex structure which helps in maintaining the catalytic activity of the different compounds. So how the enzymes are acting? So what is the activity or what is the process that is involved in the enzyme catalytic activity? So we are not able to give a single theory which will support how specificity or the activity of the enzyme is usually coming into existence. So there are different opinions that are present, and there are different ideas that can be utilized for this particular process. So all these ideas that have come into existence have basically been identified by the different experiments that have been carried out. So based on the number of experiments, based on a particular experiment, we have decided that this is how the enzyme's activity is coming into existence. So certain techniques like the X-ray crystallography, spectroscopy, or the electron spin resonance. So these have been generally utilized to identify what is the most common methodology by which the catalytic activity of an enzyme comes into existence. So the most probable cause that we see is that the enzyme has a specific location which we are calling it as an active site. So wherever their active site is available, the substrate or the reactant material that is there. So the substrate, which is also the reactant material that is there, it will come in contact with this particular active site that you have here. So the substrate material that is there, it will come in contact with this active site and it will bind at that particular active site, forming a new material which we call it as the enzyme substrate complex. So this new material that is formed is basically an intermediate material between the enzyme or the substrate and the product material. So substrate is your reactants and finally from there you are getting the product material. So if you look at this energy versus the reaction progress graph that we have, so if you see initially without any catalyst material, if you look at, this would have been the energy that was required for the process to run. So by adding the enzyme, what happens is that the energy required for the process has come down considerably. That is basically implying that by the addition of the enzyme material, it acts as a catalyst, thereby reducing the energy that is required for the process. So by the substrate material, it shall combine with the enzyme, forming the enzyme substrate complex, and the reaction will come into existence or come into completion. And once the reaction has come into a completion, then the product material that is there, that will be formed and it will be separated out from the enzyme materials. So this is how usually the substrate and enzyme reactions are taking place, forming the product materials. So the one thing that you'll have to look into is that enzymes which are acting as a catalyst will neither take part in the reaction, or there will not be any conversion of that particular material or the enzyme. So catalysts are those materials which help in increasing the rate of the reaction without directly getting involved in this process. So here also the enzyme, there is no direct involvement of the enzyme, only that a small amount of the active site that is there the substrate material is getting linked, and then the reaction is taking place at that particular active site and the product material is getting separated out from the enzyme substrate complex. Right? So based on these ideas, so based on what is the enzyme and based on what is the enzyme substrate complex and how the direct reaction is going to take place. So now we'll look into how the simple kinetics of an enzyme is coming into existence. So here what we'll be looking at is how a single product substrate material is going to get converted into the product material. So the reaction that is usually going to take place is something like the substrate material is getting converted to a product material. So as I was telling, enzymes do not take part directly in the reaction, but it will help in moderating or varying the rate of the reaction. So thereby your substrate material, whatever is there, that is getting directly converted into your product material. So based on this direct reaction that is taking place, if you look at it, so the reaction rate that we have is basically given as V, which is nothing but the rate of reaction is nothing but minus of ds by dt, and which is also equivalent to the dp by dt. So the negative sign on the ds by dt indicates that the substrate material is getting consumed. That is, there is a reduction in the substrate material, because of which you have the negative sign. And similarly, dp by dt has a positive sign because the product material, whatever is there, it is getting produced. So as long as the product is getting produced, it will increase in quantity, and the substrate is getting consumed, so it will decrease in quantity. Thereby, the negative sign indicates that the overall the reaction is or the rate of reaction will be a positive aspect. So coming to the units of the V, the units of this particular reaction or the simple enzyme kinetics, if you look at it, it would be moles per unit volume per unit time. So if you look at the aspect of the reaction rate, so what we would like to say is that the reaction rate is basically an intensive quantity. So if you look at intensive quantity, basically implies that it doesn't depend on the size of the material or the size of the compounds that is involved in the process. So there is something called an intensive property and an extensive property. So intensive properties are those properties which do not depend on the size of the material, whereas extensive properties are such kind of properties which depend on the size of the material. So for example, temperature is an intensive property, whereas mass is an extensive property. So similarly, if you look at it, so the reaction rate is also an intensive property which is defined at each point in the reaction mixture. So now what happens is that the reaction rate will also change with the position in the mixture. So for our assumption, what we will do is, or for the purpose of uniformity, what we will assume is that the reactor that we are taking, or the location where the reaction is taking place, we'll assume that it is a well mixed, that it is completely spatially uniform in the existence. And then to measure the enzyme kinetics, what we'll do is we'll take the following observation at time t equal to 0. So we have a time t equal to 0, that is the start of the reaction, wherein we'll take certain kinds of observations. So at that particular point of time, what we are going to observe is that first and foremost, we'll say that the mixture is well stirred. That is, the distribution of the concentration of the substrate material and the enzyme material throughout the reactor is at a fixed value. Now then, the second thing that we will look into is that it is closed isothermal vessel. What we mean by that is that there is no exchange of properties within the system, or there is no exchange of properties from the system inside to the surrounding materials. So that means now it is kind of a batch kind of a process, therefore, and moreover, since we considering it as an isothermal process, there is no more changes in the temperatures that is going to be present. Similarly, it contains a buffer, it contains something called a buffer solution, which is used to maintain the pH of the entire mixture. So, when we are talking about bio-based catalysts or biocatalysts as such, temperature and pH have a very high impact on the particular process. So, that is the reason we are going to take into consideration the isothermal process as well as the fixed pH value by using a buffer solution. So, there are different methodologies by which we can calculate the reaction rate. So, it can be either a spectroscopic method, it can be a manomeric method or a polarimetric method, or simple method of analysis can also be utilized to determine the kinetics that is involved. So, for this particular entire process, we will be using something called the initial rate data. So, whatever initially we have taken into account, based on that particular value, we will determine how the process is going to vary. Right? So, coming to our derivation, so if you look at it at first at time t equal to 0, that is at the start or just before the start of the process, what we are going to tell is that the concentration of the enzyme, we are taking it as E naught. So, E equal to E naught, basically implying that the total concentration of the enzyme that is present in the free and combined form is taken as basically E naught. Okay. And then initial concentration of the substrate material, we will take it as S is equal to S naught. And then initially, since there is no product material that is present, we will take the concentration of the product material as P equal to 0. So, if you observe from the graph on the right hand side, you can see that although at the initial point you see that there is some amount of the product material that is being produced, that is basically because we have not taken the actual time as the time equal to 0. So, once the material or the enzyme and the substrate are added into the mixture, there is a possibility that immediately itself some amount of the product material is being formed. So, that is the reason that we initially have a small amount of the product material that is being produced. So, this is just an assumption. So, we can use this assumption and tell that it is almost somewhere equivalent to 0. So, based on that, we will be able to calculate the kinetics that is involved in this particular process. So, the initial concentration as well as the enzyme activity, whatever we have, these can be easily reproduced, and that is why we are using these two quantities as the main process for the entire systematic development of the kinetics that is involved. So, there is a, when you are considering the in vivo studies, there is a possibility that the enzyme denaturization might take place. So, the denaturization of the enzyme is a typical quantity, that means that the enzyme might lose its activity, but that in most of these cases we will not be taking that into consideration. But if you are talking about an in vitro study, so wherever the enzyme is taken out of its original concentration or the original cell, then there is a possible of enzyme denaturation. So, most of our analysis we will be doing it in vivo analysis, so that there might not be an enzyme denaturization that is present. So, finally, when we are calculating, we will be estimating the units of enzyme. So, the units of enzyme, or the enzyme activity is usually designated as the amount of enzyme which gives us the certain amount of enzyme activity. So, we will be calling it as international units or just units, so as to represent the enzyme's activity. Right? So, the rate of reaction is initially basically a first order reaction at the relatively lower concentration. So, if you look at the graph itself, you can see that at lower concentrations of the material that is there, or the substrate material that is there, you will see that the enzyme reactions or the reaction rate that is involved in the process is basically a first order reaction that is taking place. So, as the concentration is going to increase, what usually happens is the reaction order shifts from the first order reaction to the zero order reaction. So, based on this, what we can tell is that the rate of the reaction is directly proportional to the amount of enzyme that is present in the mixture that we are calculating. So, the higher the amount of the enzyme that is present, the higher the rate of reaction that is present. So, basically again implying that since the enzyme that is present is acting as the catalytic material, thereby the rate of the reaction that is involved in the process is going to increase based on the amount of the enzyme that is present. So, these are some of the observations that we can make on basis of the rate equation that we have determined. So, from this what we can say is that V equal to Vmax into S by Km plus S, which is the rate of reaction or the rate equation for this particular process. So, this particular process we will call it as the Michael-Menten kinetics or Mikhail-Menten kinetics. So, based on our analysis, it will be Michael-Menten kinetics or Mikhail-Menten kinetics. So, this would be the final equation that we will come across, where we will be able to calculate the rate of reaction that is involved. So Vmax, which is nothing but the maximum rate of reaction, is something like a quantity if we call it as alpha into E naught, where E naught is the initial concentration of the enzyme that is being added into the system. So coming into how we are going to derive this particular equation, that is V equal to Vmax into S by Km plus S. So there is a two-step process that is involved in this reaction. So the step one, what happens is that the enzyme and the substrate will combine to form an enzyme substrate complex. So that is your first half of the reaction, or the first step that is present in the reaction. And from that, once the enzyme substrate complex is formed, the second step is that the complex will dissociate to give you the original enzyme, which is the catalytic material, and the final product material that is present. So these are the two steps that is involved in the conversion of the substrate material to the product material by using the enzyme as the catalytic compound that is present. So from the step one, what we can say is that the substrate material will combine with the enzyme to form the enzyme substrate complex, which is basically a reverse reaction. So the rate of forward reaction, or the rate constant for the forward reaction, we are taking it as K1, and the rate of backward reaction, we are taking it as K minus 1. Right? So the step 1, wherein the substrate material is getting combined with the enzyme to form the enzyme substrate complex, the rate constant for the forward reaction is K1, and the rate constant for the backward reaction is basically K minus 1. And in the second step, what we are observing is that the enzyme substrate complex is dissociating to give you the original enzyme, which is the biocatalyst. Along with that, there is a product material that is being formed, and the rate constant for this particular step is basically given as K. Right? So based on the reaction from step 1, it can be assumed that at equilibrium concentration, what we can say is that the concentration of the substrate material into the concentration of the enzyme divided by the concentration of the enzyme substrate complex is basically given as K minus 1 by K1, which we are taking it as a dissociation constant, which we call it as Km. Right? This is for only the step 1. That is, if at an equilibrium condition, that is the rate of forward reaction will be equal to the rate of backward reaction, then from this scenario, what we can say is that the concentration of S into the concentration of E, which is nothing but the concentration of the substrate material into the concentration of the enzyme, whole divided by the concentration of the enzyme substrate complex, is basically given as K minus 1, which is the rate of backward reaction by the rate of forward reaction, which we are calling it as the dissociation constant. What we know is that the step 1 is a reversible process, whereas the step 2 is an irreversible process. So when the step 2 is an irreversible process, we are taking that to determine the rate of the reaction for the entire process. So the rate of reaction V equal to dP by dt is basically given as K2 into concentration of ES, which is nothing but the concentration of the enzyme substrate complex. Right? So all the available enzymes, whether it is in the free form or whether it is in the combined form or a complex form, whatever the enzyme material that is available, it will always be summed up and equal to the original or the initial concentration of the enzyme. So when you look at it, E is the amount that is freely available, and ES is the amount that is available in the combined form as the enzyme substrate complex. So sum of, or the amount of these both, or the amount of enzymes in both these concentrations will be equal to the initial value, which is given as E naught. Right? So based on the mass balance for the substrate and enzyme complex, what we can write is that the rate of reaction V equal to minus ds by dt. So from the first reaction, we can say it as K1 into concentration of S into concentration of Sc. This is nothing but for the forward reaction, minus of K minus 1 into concentration of Es, which is for the backward reaction. And from this, what we can say is that with respect to the change in the enzyme substrate complex, that is dEs by dt, we can say that it is K1 into concentration of S plus concentration of E into E minus of K minus 1 into K2 and minus of K minus 1 into concentration of Es minus of K minus K2 into concentration of EH, which we combine together to form this particular equation. So this is the rate of substrate consumption and this is the rate of your enzyme substrate complex changing with respect to your time that is involved. Right? So from the above expressions, we have come to a generalized idea of how much amount of the concentration of the substrate is changing and how much amount of the enzyme substrate complex is being formed. So for all this particular process, if you take into consideration, what we are telling is that the initial conditions or the initial condition for all the above expressions is that the substrate material at a condition of S is equal to 0, we are taking it as S0, and initially there is no enzyme substrate complex, only the substrate material is available and no reaction has taken place, because of which initially the concentration of the enzyme substrate complex will be equal to 0. So based on this above condition, what can be estimated is that the rate of change of enzyme substrate complex at the initial condition is also going to be equal to 0, which we will consider as a very good approximation based on our initial condition. So by taking this into consideration, that is the third equation that we have, that is d of concentration of Es by dt is equal to K1 concentration of S into concentration of E minus of K minus 1 plus K2 into concentration of Es. So from this, what we are telling is that the left hand side of the equation, that is this particular expression, will be equal to 0. So based on that, we are converting it into this particular expression, and then further by exchanging the or by bringing the negative term to the right hand side, we are telling it as K1 into concentration of S into concentration of E is equal to K minus 1 plus K2 into concentration of Es. So from this, we are determining the original value of the concentration of Es. That is basically calculated as K1 into concentration of S into concentration of E by K minus 1 plus K2, which what we are going to do is we are going to substitute it into the initial half of the expression. Right. So now looking at the original value that you have, that is the right hand side, K1 SC into is equal to K minus 1 plus K2 into concentration of Es. So what we are telling is that, see, if you remember, we are told that the concentration of the enzyme in its combined form or in its free form will be equal to its original value. So from that particular assumption, we are telling that the concentration of Es, we are changing it as concentration of the initial value minus the enzyme that is available in the free form. That is, the concentration of enzyme in the substrate complex is equal to the initial value of the enzyme that is available minus the concentration of the enzyme that is available in the free form. Right. So again, further we are going to expand this particular expression. So we have K minus 1 plus K2 into E naught minus of K minus 1 plus K2 into E. So the second term that we have, that we are taking it to the left hand side. Thereby we are taking it as K1 concentration of S into concentration of E plus K minus 1 plus K2 into concentration of E is equal to the right hand side we have K minus 1 plus K2 into concentration of E naught. So from here we are calculating the concentration of E. So from both these terms, we will be taking the term E as common, that is the concentration of the enzyme in its free form, we are taking it as common, and from there we are getting it as K minus 1 plus K2 into concentration of the enzyme in its original or the initial condition is E naught, and from the left hand side we have K1 into S plus K minus 1 plus K2. Right? So from these left hand side of the expression, we have taken the value of E as common from these both terms. The terms remaining are K1S and K minus 1 plus K2, which we are taking it as the denominator in this particular equation. Right. So now we have the rate of equation V, which is given as minus ds by dt. So this is the expression that we have with respect to the substrate material. So with respect to the change in the substrate concentration, this is the particular kind of equation that we have, that is K1 into concentration of S into concentration of E minus of K minus 1 into concentration of Es, which we have taken it from the first half of the irreversible mixture. And from this, what we are telling is that we are replacing the value of concentration of Es by the value which we have got from this particular equation. So this value that we have obtained, that we are going to replace in place of the concentration of Es. So from here, if you see, K1 into concentration of S into concentration of E minus of K minus 1 into, instead of concentration of Es, we are writing it as K1 into Se by K minus 1 plus K2. So this particular expression we are going to replace as in the particular equation. So further, we are going to expand this particular equation. So if you look at it, we have K1 SC and K1 SC on both these terms. So we are going to take that particular term as common, and then we have 1 in this particular term, and then from here we have minus of K minus 1 by K minus 1 plus K2. So once you do the common denominator, so you have K minus 1 plus K2 minus of K minus 1. So that particular terms, if you look at this, so the K minus 1 terms are going to cancel out in this particular equation. So if you look at it, this particular expression that we have, it will be only this particular form. We will see it is K minus 1 plus K2 minus of K minus 1 whole divided by K minus 1 plus K2. So if you remember, these two terms are getting cancelled out. So we have only K2 in the numerator. So when we write that, so we have K1 concentration of S into concentration of E, and the only term that we have is K2. So this particular expression will look like K1 into K2 concentration of S into concentration of E by the denominator value we have K minus 1 plus K2. So that is the rate of equation with respect to the substrate concentration that we have. right? So once we have come up to this equation, now again we will be able to substitute the value of E that we have taken from this particular expression. So this is the value of E, or the concentration of the enzyme that is available in its free form. So that particular value you are going to substitute in place of this particular expression here. So what I have done is basically K1 into K2 into concentration of S by K minus 1 plus K2. I have separated it out into the concentration of E. So that particular value, that is K minus 1 plus K2 into concentration of the enzyme in its initial condition E0 by K1S plus K minus 1 plus K2. So this is the entire expression that we have with respect to the entire equation. So if you look at this particular terms, K minus 1 plus K2 and K minus 1 plus K2, these two terms are getting cancelled out. And now the expression that is remaining is K1 into K2 into concentration of S into concentration of E0 by K1S plus K minus 1 plus K2. Now, for our further analysis, what we'll try to do is we'll divide the numerator as well as the denominator by the term K1. So K1 is getting cancelled out in this particular term, and then in here also K1 is getting cancelled out, and K minus 1 plus K2 by K1, that we are considering as the overall equation. So now from this new expression that we have, what we are going to do is we'll take these two components, that is K2 into E0, that is basically equivalent to the Vmax value, and along with that we have the term S, and this particular expression, that is K minus 1 plus K2 by K1, that we are calling it as the new dissociation constant, which is K. So using this, what we have finally found out is that V equal to Vmax into S, Vmax into S whole divided by Km plus S. So this is the final form of the Michael-Menten kinetic equation that we have derived using the simple enzyme kinetic model. So using the simple enzyme kinetic model, what we have taken is that the enzyme is combining with the substrate material and the reaction is taking place at the active site. Once the reaction is getting completed, the product material is dissociating from that particular reaction. So the first step, that is the combination of the substrate material with the enzyme, that is a reversible process. That is, the enzyme substrate material will be formed, as well as the enzyme substrate material can go back to its initial form. The step 2, which is nothing but the dissociation of the enzyme substrate complex into the product material, that is an irreversible step, and based on that we have derived the set of, and based on the initial assumptions that were there and based on the initial conditions that were there, we have derived and we have found out that the overall equation or the rate expression for the simple enzyme kinetics involving one substrate material is given as V equal to Vmax into S by Km plus S. So this is basically considered as the final form. Sorry about that. This is basically considered as the final form of the Michael-Menten kinetic equation. So based on this Michael-Menten kinetic equation, what we have to find the different Michael-Menten parameters. So it is not quite easy to estimate the values of Vmax, which is the maximum rate of equation, as well as the value of Km, which is nothing but the dissociation constant, by using this direct equation. So the direct equation that we have, V equal to Vmax into S by Km plus S. To determine the values of Vmax and Km directly using this expression is a little difficult. So for that particular reason, what we are trying to do is we are trying to bring about some changes in this equation so that we are able to calculate the values of the expression without any difficulty. So for that we will be using three different processes. The first process that we will come across in this particular is something called the Lineweaver-Burke equation. So the Lineweaver-Burke plots. So for this, what we will be doing is, so we have the equation as V equal to Vmax into S by Km plus S. So first thing that we will do is we will invert this reaction. So taking the inversion, what will happen is we will have 1 by V equal to Km plus S by Vmax into S. So if you look at this, if you separate out the expression, we have 1 by V, and the first half is Km by Vmax into 1 by S plus S by Vmax into S. So S gets cancelled out. So we have only the term 1 by Vmax. So now this particular equation, if you look at it, is in the form of y equal to mx plus c. So if you consider y as 1 by v and x as 1 by s, so in this particular case, we will get the slope of the equation as Km by Vmax and the intercept value C, that will be equal to 1 by V max, right? So this is how we are going to get the expression for Km and V max on this particular term. So if you are able to plot a graph of X versus Y in this particular case, the value of 1 by V versus 1 by S, and if you are able to predict, then you will be getting a straight line for that value of 1 by V versus 1 by S, and that slope of that particular straight line will give you the value of Km by V max, and wherever the intercept is formed, wherever the line is intercepting with the Y axis, that particular value will give the intercept, which is nothing but 1 by V max. So based on that particular value, initially you will find the value of V max, and using that V max value, you will substitute it into this particular equation here, and using the slope of that particular equation, we will calculate the value of Km, which is the new dissociation constant. So this method of calculating the two Michael-Menten parameters, that is by using the graph of 1 by V versus 1 by S, that particular methodology is basically called as the Lineweaver-Burk plots. The second method that we will be utilizing is whatever we have done. Similarly, on inverting similar kind of a condition, we will invert the expression initially. So 1 by V equal to Km plus S by V max into S. So we will be getting it as 1 by V is equal to Km by V max into 1 by S plus 1 by V max. So now we know the concentration of the substrate material throughout the entire system that is available. So what we will do is, we will multiply the entire reaction that is there, or the entire equation that is there, by the term of the substrate concentration, that is S. So in that particular case, we will have S by V is equal to, so here the S terms are getting cancelled out. So Km by V max plus S by V max. So still the equation is of the form Y equal to Mx plus C. The only thing is that here the value of Y is concentration of S by constant, or the rate equation B, and the X in this particular term is the substrate concentration, that is S. The slope M will be given by the slope M will be given by 1 by V max, and the intercept, that is C, is basically Km by V max. So when you draw a graph of S by V, so your Y coordinate in this particular case is the concentration of the substrate material by the rate of the reaction, that is S by V, versus the concentration of the substrate material. In that particular case also, you will be getting a straight line which will intercept the Y axis at a particular point. So from this straight line, the intercept value will give you the term for Km by V max, and the slope of that particular expression will give you the value of 1 by V max. So initially you will use the slope to find the value of V max, and using that value of V max, you will substitute that value of V max into this. You know the value of the intercept, that is C, and using that you will calculate the value of Km as C into V max, thereby giving you both the values of V max and Km, which are the Michael-Menten parameters in this particular scenario. So the plots that are most commonly used, or the plot that we are developing in this particular case, that is S by V versus S. This particular plot is basically called as the Haynes-Wolff plots. So by using the concentration of the substrate material, or the ratio of concentration of substrate material with the rate of reaction with respect to the concentration of the substrate material, the plots that you develop in this particular case, we will call it as the Haynes-Wolff plots. And finally, the last method, or the third method that can be utilized to determine the Michael-Menten parameters, that is Km and V max. So again, coming back to the original equation, that is V equal to V max into S by Km plus S. So I'm just interchanging the expression. So we get V into Km plus S is equal to V max into S. So from this, we are bringing V max into S to the left hand side, and then Km into the Km is into the expression as such. So we have V max into S is equal to V minus V max into S, that is equal to Km into V. So V max into S, V minus V max, the entire term for the S on one side, and then we have the other term that is remaining, that is Km into V. So from this, we are calculating as V minus V max is equal to minus of Km into V by S, that is the common slope that is available. So here you do not have an intercept, and finally we are taking it as V equal to V max minus of Km into V by S. So again, here also it is of the form Y equal to Mx plus C, So the mx plus c, in this particular case, we have the y is equal to the v and your x is equal to v by s. That implies that the slope is equal to minus of the km, and the intercept that you have, it is basically nothing but v max. Right? So using this particular expression, or by using this particular equation, you will be able to find out the values of km and v max directly. So you do not have to represent, or you do not have to replace any of these components. But by using this particular scenario, you will be able to calculate the values of Vmax and Km directly without any interference. The slope will give you the value of minus Km, and the intercept at this particular point will give you the maximum rate constant, or the maximum rate of reaction that is involved in this process. So using this, or the final methodology that where we have determined the value using V at the rate of or the rate equation with respect to the rate equation and the substrate concentration. So this particular scenario, we are calling it as the Eddy-Hofstee plots. Right? So these are the three methodologies by which you will be able to calculate, or by which you will be able to evaluate your Michael-Menten parameters. So you have the Lineweaver-Burk plots, then you have the Haynes-Wolf plot, and finally you have the Eddy-Hofstee plots. So these are the three methods by which you will be able to evaluate the Michael-Menten parameters that you have developed by using the Michael-Menten kinetics, which will give you an understanding of how the dissociation constant and the maximum rate of reaction is calculated from each of the individual terms that is present within the expression. Right? So with this, I would like to conclude the topic of kinetics of the simple enzyme catalytic reactions. So if you have any further queries, I guess you can get back to me on the details. So you have my contact details, so you will be able to get back to me on any of the queries that you have with respect to the kinetics of the simple enzyme catalyzed reactions. Thank you everyone for your patience. Thank you very much.
Like, share and subscribe. Hit the bell icon for more updates.